Today, I want to talk about some exciting data we have looking at the hERG potassium channel. I'll talk a lot about what hERG is for those who don't know and what we do know about it. But the really exciting stuff about our work here is, I think, the new idea that a piece of the hERG potassium channel goes inside the nucleus and seems to be regulating gene expression in there. I'll start out and I'll give a background of what we know about hERG1 and what it does in cardiac and neuronal electrophysiology and kind of what the differences are in functionality there. And then I'll dive into our stuff with this piece of the channel that goes inside the nucleus and some of the work that we're doing to try and figure out what the targets of hERG1P function are in cardiomyocytes. So for those not familiar with cardiac electrophysiology, the contraction of the heart is regulated obviously by an action potential, and that action potential takes on many different forms as it moves from the sinoatrial node and down through the atria into the ventricles, and that difference in shape or morphology is driven by sometimes subtle and sometimes dramatic changes in the expression profile of the ion channels that underlie the action potential. Some entire ion channel forms will be lost, or it can be more subtle differences like shifts in the subunit abundance of those individual channels. And so my lab is primarily focused on ventricular repolarization. So the cardiac action potential in ventricular myocytes is very long. It's very distinct from what you might see in a neuronal action potential. So instead of 1 or 2 or 3 milliseconds, we're dealing on the order of 100 to 400 milliseconds. So the time scale is somewhat unique from what you see in the brain. And then so the current that we focus on is something called IKR, which represents a rapid delayed rectifier potassium current. So I study hERG, which is the ion channel that conducts this current. Now we know a fair bit about the role of human IKR and hERG in adult repolarization, but that story is less complete when you start applying to early development and maturation of cardiomyocytes. In the adult myocardium, it's a very straightforward effect where a loss in IKR or hERG current leads to a prolongation of the action potential, and that drives an increase in cardiac arrhythmia. In developing myocytes, the electrophysiology is distinct, and it's dynamic through the duration of human development. This is a slide taken many years ago, actually, from embryos of a chicken. But the concepts here are the same across species, where early in development, the ventricular action potential is very depolarized to what you see in adult myocardial tissue, with resting membrane potentials that can be as high as minus 30 millivolts, which is 50 millivolts more depolarized than what we see in adult myocardium. But during development, the resting membrane potential is hyperpolarized. The magnitude of the action potential becomes larger, and you see these changes also manifest in the behavior of individual myocytes, whereby cardiomyocytes taken from embryonic and even some fetal tissue spontaneously fire their own action potentials, whereas if you took the ventricular myocytes from an adult myocardium, they're quiescent and only fire an action potential when stimulated. So we know from numerous studies over the past 50 years that the spontaneous action potentials in developing myocytes is caused by an almost complete absence of a hyperpolarizing rectifier current IK1. These myocytes also have minimal sodium currents, and so it's almost like a simplified system where you have a calcium current driving the upstroke, and then you have the presence of IKS and IKR, which are repolarizing currents, the rapid delayed rectifier, which we study, and the slow delayed rectifier, which other groups focus on. But in this system, you can imagine, because there's a different complement of channels, that the effects of disrupting IKR might not necessarily match what we see in adult myocardial tissue. And so some of that has been borne out more recently with the advent of stem cell-derived cardiomyocytes, which are immature, but if you're focusing on mechanisms of sudden death in the young, it's actually you could think of it as an advantage. So generally speaking, in adult cardiomyocytes and a subpopulation of stem cell-derived cardiomyocytes, if you block IKR, if you block the hERG current, you get AP prolongation and something that we call early after depolarizations, which are a cellular manifestation of cardiac arrhythmia. So here we're using something that's a very selective blocker for IKR, E403 those action potentials get longer, and we get those EADs in this population of stem cell-derived cardiomyocytes. You can also see the spontaneous firing that's going on here. So these are not paced, these are ventricular myocytes that are beating on their own. But what's surprising in this scenario is that because of the lack of IK1, the majority of cells, when we block IKR, we actually get a depolarization of the resting membrane potential and action potential shortening. And so this is sort of like the simplest way to demonstrate that the role of IKR is dependent on the maturation state or developmental state of the cardiomyocytes. And that's really where my lab resides and is studying it. So we use stem cell-derived cardiomyocytes, and they're, as a model of the developing myocardium, to look at the effect of disrupting hERG on electrophysiology and cellular physiology at distinct stages of development. And one of the big drivers that we do this is that sudden cardiac death is about 6 times more likely in the first year of life than any other time in childhood or adolescence. And when you really think about that, it's surprising when we put in the context of how much value we put on an infant's life. We actually know very little about the physiology in that early stage of life. And so stem cell-derived cardiomyocytes in their because we're able to make them continuously, really represents a new frontier, I guess, in our ability to model the earliest stages of electrophysiological development, and try to understand what makes an infant so susceptible to sudden death. So now I just want to dive in a little bit more about what we do know about hERG channel biophysics and its role in human physiology. So hERG, which stands for the human ether-go-go-related gene. I'm not going to talk about where that name comes from, but you can always email me and I'll give you the full backstory. But it's a fun name. It's encoded by KCNH2. And in the heart, we know that it conducts IKR, the rapid delayed rectifier potassium current, and a loss of IKR, either through loss of function variance or off-target pharmacological block, predisposes individuals to cardiac arrhythmia and sudden cardiac death. But KCNH2 is not limited to the heart. It's also expressed almost ubiquitously throughout the body, but it's particularly present in the brain and pancreas. And it's associated with a number of causes of death that may or may not be linked to its role in cardiac repolarization. And that includes sudden infant death syndrome, intrauterine fetal death, and sudden unexplained death in epilepsy. So this is two subunits of hERG facing each other. This is a cryo-EM structure from Rod McKinnon's group some years ago. And so you can see this sort of standard 4 helical segments forming a voltage sensor domain, and then an S5 and an S6 along with the P loop to form your potassium selective pore. In hERG, you also have some somewhat complex cytoplasmic domains. Off the N terminus is para-ARNT-SYM domain, which is found in all KCNH channels. That interacts with a cyclic nucleotide binding homology domain of the C terminus to regulate, I guess you could say, the communication between the voltage sensor and the pore. And then we have some fairly disordered protein downstream of the CMBHD. That's actually going to be the focus of most of our talk once we get there. Its role isn't particularly well understood. So hERG has unique gating characteristics in that its time course of inactivation is orders of magnitude faster than its time course of activation. So they have a relatively slow activation compared to inactivation. What that means is that as the channels move from the closed to open state, they almost immediately become inactivated. And the way that manifests physiologically is that the currents are suppressed at depolarized potential. So if we hold at -80, this is just HERG1A expressed in HEK cells. We depolarize the membrane. You can see the channels begin to activate, but then quickly that current is suppressed by the transition into this inactivated state. If we step back down to a negative resting membrane potential, the channels quickly recover from their inactivated state, but because the secondary transition from open to closed is slow, they reside in that open and conducting state. And so you get actually an increase in current at a more negative potential, even though you've reduced the driving force for potassium. So you can see that that profile during a ventricular action potential waveform. So if we depolarize the membrane, the channels activate but quickly are suppressed at what we call the plateau phase of the action potential. And it's not till later in repolarization that the channels begin to recover from the inactivated state. But they reside in this open state during the final phases of repolarization. So you get a bolus of current during phase 3, the final phase of action potential repolarization. So we spent a lot of time paying attention to tail currents, the current from HERG channels that occurs during the tail end of an action potential. The role or the contribution to electrophysiology and excitability of HERG varies very widely between cardiac and neuronal tissue. So in a ventricular myocyte where your action potential can be up to 400 milliseconds long, depolarization is long enough to have a very large number of HERG channels open during that time course. And so that population of channels will open and then drive repolarization of each individual action potential. If you have a loss of IKR from pharmacological block or a loss of function variant, you get a reduction in IKR. That corresponds to an increase in the duration of each individual action potential. And that's what contributes to arrhythmia and sudden death in the adult myocardium. But that's very different in neuronal tissue where each action potential is so short that it's really hard to quantify the number of channels that are pushed towards their open or activated state during an individual action potential. But what does happen is over sustained stimulus, over multiple action potentials fired, you do accumulate a population of HERG channels in the open state, and this helps to dampen excitability through a process called spike frequency adaptation. So if you block HERG in a neuron, you will have a reduction in the HERG current, and you lose spike frequency adaptation. So you'll have an increase in excitability, but you won't really see any change in the duration of the individual action potential, but you will see an increase in excitability. So that kind of just highlights the big sort of broad stroke differences between neuronal and electrophysiology regulated by HERG. Now, HERG is a multimeric, heteromeric ion channel. We know of at least 3 subunits that can combine to form a functioning HERG1 potassium channel. HERG1A, HERG1B, and 1C. And they all come from the same gene, just with alternate start sites for each individual transcript. So the full-length channel, the one that we know the most about is HERG1A. So it's encoded by these 15 exons here. And the first 5 of those exons encode a parent SIM or PAS domain that I mentioned earlier, regulates the communication between the voltage sensor and the pore. We have 2 other subunits, 1C and 1B, that have their own promoters that start downstream of 1A. Now, what's interesting about these, both 1B and 1C, they create truncated and unique N-termini that lack a fully functional PAS domain. And that turns out to be really critical in fine-tuning the kinetics and magnitude of IKR in different tissues. For the purpose of our talk today, I'm going to focus on 1A and 1B because those are actually the only two subunits that are found in cardiac tissue. 1C is a primate-specific neuronal channel that's associated with schizophrenia. There's also this guy right here, which is HERG-USO. This is an extension of exon 9 and actually forms a subunit that will heteromize with the full-length channels, but then is retained within the ER, so it doesn't actually traffic to the membrane. We're not really sure what HERG-USO does just yet, but it is there. So HERG channels like to be heteromers. Here's the cryo-EM structure again from Rod McKinnon. You can see the voltage sensing domains in yellow, excuse me. The pore in green, and then the PAS domain is in orange. CMBHD is in blue or cyan. They interact tightly here, and then through an extension here called something called the PAS cap that helps regulate the communication between the VSD and the pore. I told these guys that I didn't have any videos. I guess I do have videos. I guess I'm cool enough for videos. Now the other subunit, HERG1B, has a short and unique N-terminus that lacks a PAS domain. This isn't what it really looks like. We don't know what it actually looks like. This is what we think it looks like, but we don't have a cryo-EM structure. We just lop it off. And we also don't know the stoichiometry. So I'm sure that there's someone in the group wondering what the stoichiometry is. I'd love to know the stoichiometry, but we haven't been able to peg that down yet. We think that it's dependent on the physiological state and the developmental state of the tissue that's the channel resides. In any case, the absence of this PAS domain in the heteromeric channels causes a twofold acceleration of the time course of activation, deactivation, and recovery from inactivation. And that leads to about a twofold increase in the amount of current that is conducted during a ventricular action potential waveform. So an extension of that is that since the 1B subunit does not have a PAS domain, you could think of it as the PAS domain of 1A suppresses herd current. And we have more and more evidence that the relative abundance of 1A to 1B, or sort of the relative amount of PAS domains, is used as a tool by the cells to fine-tune the shape, magnitude, and timing of IKR in cardiomyocytes. So we can target the PAS domain either directly or indirectly to modify the magnitude of IKR and change the kinetics or the speed of repolarization. In this experiment, this was done in stem cell-derived cardiomyocytes, and we're recording action potentials in rapid succession, paced at 1 Hertz. Here's a vector control, so you can see the action potentials repolarize fairly uniformly. But then if we use an shRNA to disrupt selectively HERG1B, we get a reduction in the magnitude of IKR, and we get an increase in action potential duration, and we get this increase in beat-to-beat variability. Both of those are markers of proarrhythmia. But you could make the argument that we're knocking down protein there, so you just have less channels at the membrane. So a more elegant experiment is actually to mask the behavior of HERG1B. And we do this by overexpressing the N-terminus of HERG. So we're overexpressing the PAS domain, and then that PAS domain can fill this empty receptor site made by the unique N-terminus of HERG1B. So we do that, we transform the gating of the heteromeric channels in the cardiomyocyte to something that's indistinguishable from a HERG1A homomeric channel. So we get smaller current from the same number of channels, and lo and behold, we get an increase in action potential duration and an increase in beat-to-beat variability. So that tells us that not just the number of channels at the membrane, but like the composition, the relative balance of 1A to 1B subunits is important for normal repolarization in human cardiomyocytes. We can do it the other direction too, where using an antibody, in this case, it's a single chain variable fragment antibody that binds to the PAS domain and inactivates it, prevents it from binding to its partner with the CMBHD. We accelerate gating, we cause an increase in the magnitude of IKR, and that subsequently accelerates repolarization. So we can target this interaction between essentially the PAS domain and the rest of the channel to modify IKR and cardiac repolarization. So that wraps up the background on hERG that I want to talk about and brings us to this new concept, which is a polypeptide encoded by casein H2 that is targeted to the nuclei of immature cardiomyocytes. So when I was setting up my lab, and we were revalidating antibodies that we had used a million times before, some of you may discover this soon, where you start your own lab, and things that worked just fine in your PI's lab before, for whatever reason, maybe it's too humid or whatever, but things shift. So you want to make sure that everything still works in your hands. So we're validating these antibodies, and we one against a P-loop. These are stem cell-derived cardiomyocytes. Labels just as we'd expect. We see enrichment here in the sarcoplasmic reticulum, which is the cardiac version of an ER. And then you can see it distributed through the cytoplasm and out to the surface membrane. That's what we expect. We've labeled the nuclei with DAPI. But then when we label the very end of the C-terminus, we were really surprised in that the majority of our immunofluorescent signal was actually inside the nucleus. And it's not something we had seen in HEK cells. And at this stage, we thought that maybe this was just bad antibody behavior. There was something inside the cells that was cross-reacting with the antibody. But following the data, as one can often do, we did some follow-up experiments. So stem cell-derived cardiomyocytes are really immature, especially using sort of standard practices where the cardiomyocytes are maybe 15 to 30 days old. They probably resemble something found in a late embryo or early fetus. So we use some of our own tricks to push the cardiomyocytes down their maturation pathway. This is something called Matrix Plus, but we see a similar effect no matter what technique that we use. So we mature these cardiomyocytes, so they're more like a late fetus, maybe first day neonate, like maturity. And we can deplete the amount of immunofluorescent signal inside the nucleus. It's still significantly different from our P-loop. So there is a little bit more enrichment going on here, but we can control it to a degree. So that was kind of interesting. So whatever we're seeing is developmentally regulated, at least in the cardiomyocytes. But then we turned to some native cardiomyocytes from rat with the idea that maybe what we're seeing is an artifact of in vitro differentiation. And there, we were very surprised. So we still see some enrichment in the neonates that is comparable to what we saw with our Matrix Plus in vitro maturation. But then in our adult myocytes, the difference between distribution of our antibodies goes away. So basically, whatever we're tracking at this point, this nuclear polypeptide is not present or not significantly enriched in the cardiomyocytes of an adult, or the nuclei of an adult cardiomyocyte. But it does suggest that we have perhaps a piece of the channel, not the whole channel, but a piece of the channel that's targeted to the nucleus in the earlier stages of development and maturation. But as I mentioned, antibodies can be dirty. They can pick up on all sorts of things. So we generated a casein H2 null line of stem cell-derived cardiomyocytes. So we put a 2 base pair insertion in exon 5 to basically knock out all of the possible subunits of hERG that could be transcribed. And then we differentiate those stem cells into cardiomyocytes. Here's our wild type isogenic control. You can see there's plenty of hERG there and lots that overlaps within the nucleus. But then our casein H2 null, that immunofluorescent signal is all but abolished. This quantified here. Not really sure why those are shifted. So, this is encouraging. So we also we could see plenty of IKR in our wild type cells that goes away in the casein H2 null. These cardiomyocytes still fire action potentials, and they show a voltage-gated calcium current. So we know we're not just differentiating iPS cells into some weird non-cardiac phenotype. And this is pretty convincing evidence that the polypeptide that's inside the nucleus, that immunofluorescence that we're seeing inside the nucleus, is encoded by casein H2, so the gene that generates hERG. We also wanted to know how that polypeptide would get inside the nucleus, and the best studied pathway is the classical nuclear localization pathway, whereby an alpha carrion binds to a nuclear localization sequence, and then that's bound again by a beta-1 carrier ferrin, which acts as an adapter protein to shuttle things through the nuclear pore complex. So we blocked both alpha-1 and beta-1 activity using importazole and ivermectin. And in the cardiomyocytes, we're able to essentially stop the targeting of the polypeptide inside the nucleus. So that's helpful because it gives us an idea of what to look for mechanism of targeting inside the nucleus. So there should be a nuclear localization sequence specific for the classical pathway. And that's helpful too, because that's a very well-studied pathway, and we have very good predictive software for identifying those potential motifs. So when we screened the amino acid sequence of the full-length protein, we found a putative nuclear localization sequence just downstream of the CNBHD. So outside of the main structural domains of the channel, there is this nuclear localization sequence. And so we reached out to a colleague of mine, Matt Trudeau, who's had a very successful career looking at the interactions between the N and C termini of hERG using FRET. And so he has all sorts of truncates of the channel that are fused with citrines and CFPs and YFPs and all the fun stuff. I told him what we were doing. He said, yeah, I have some stuff. I don't know what you want. I said, just send me everything, and we'll see what we can make of it. So he sent us what we ended up calling the party pack of polypeptides or vectors, and just started transfecting them into HEK cells and then seeing where they went. Just as simple of an experiment as we could possibly do. So if we express the full-length C-terminus, so just cutting off just downstream of S6, this has our putative NLS. Most of the time, it sits inside the cytoplasm. Occasionally, it does get enriched inside the nucleus. About 10% of the time, we see really strong nuclear enrichment. So we think that there was some sort of cell cycle regulation going on there. With the proximal C-terminus, so this has the C-linker, the CMBHD, but not the nuclear localization sequence. It basically goes wherever the citrine wants. There's no real targeting. The average is at about 1. So that seems like a good negative control. And then when we do just the distal C-terminus, so we've got the NLS here, this unstructured region, and we don't have all of this size or baggage from the CMBHD and C-linker. This polypeptide is almost exclusively targeted inside the nuclei. So you can see the DAPI stain in red, and really beyond the perimeter of the nuclei, there's almost no immunofluorescent signal. And that kind of resembles what we were seeing in the cardiomyocytes, where the majority of our fluorescence was inside the nucleus. And then if we delete the NLS from that polypeptide, that nuclear targeting goes away. So this, to us, this told us that the NLS was a critical structural determinant in targeting the polypeptide inside the nucleus. We were also curious if it was following the appropriate path. So we blocked nuclear targeting with importazole and ivermectin again, and like what we'd seen in the cardiomyocytes, blocking these pathways would diminish the amount of nuclear enrichment, not nearly to the same degree that we saw in the cardiomyocytes, but we were still able to attenuate that enrichment. So we think that the distal C-terminus construct that we had at this point was likely a fairly good mimic of the native polypeptide. So the last thing we wanted to do was see if there was some effect on electrophysiology. I mean, we have a polypeptide that goes into the nucleus, but if we don't see any sort of physiological phenotype, what's really the point? So we went back to the HEK cells, and now we're using HEK cells that are stably expressing the full-length protein. And we transfect those cells with GFP, and we get normal voltage-dependent herd currents, which is great. And then if we overexpress this distal polypeptide, all of those currents get uniformly smaller. We see a large reduction in tail current. We see an even larger reduction in the step pulse current or the steady state current. You can see that quantitatively here. Qualitatively, it really shows up well in these family traces. So it really tells us that the distal polypeptide is likely reducing the number of channels at the surface membrane, assuming that there's not a change in selectivity, but also changing the gating behavior of the channel, right? Because more channels are getting inactivated during this step pulse here, or at least that's what it appears to be. If we normalize the step pulse to the tail current, so essentially we're getting now, we're just focusing in on that gating behavior, we can see that this reduction is still retained here. So if we control for the number of channels at the membrane, we still see a reduction in current in the presence of the distal polypeptide. So it really suggests that inactivation is being stabilized by this construct here. Now, it could still be that that is an artifact, not necessarily an artifact, but that maybe the distal polypeptide is interacting with the full-length channel directly. So we repeat this experiment with the delta NLS, and remember, that doesn't go inside the nucleus. And now all of those effects go away. The delta NLS looks just like our GFP. So that tells us that the distal C-terminus construct has to go inside the nucleus, do whatever it's got to do there, and then that feeds back to the channel at the surface membrane. So it's entirely dependent on its behavior inside the nucleus. We've done some initial follow-up experiments. You know, we also see that the distal polypeptide is slowing the time course of deactivation. And we also see this very specific effect on the time course of inactivation recovery, where the distal polypeptide is slowing recovery from inactivation. And when you look at that collectively, it really suggests that whatever the distal construct is doing inside the nucleus is affecting the interaction between the PAS and the CMBHD, because these effects are very similar to what we see when we increase the engagement of the PAS domain with the rest of the channel. So to summarize that, we have a casein H2 encoded polypeptide that is targeted to the nucleus. It binds to an alpha and a beta-1 carrier. It's targeted inside the nucleus, where presumably it interacts with the DNA to change gene expression, and that feeds back to alter the full-length channel behavior at the surface membrane. This seems to be a maturation-dependent expression profile that ultimately leads to an inhibition of current. Another thing about that is that, if it's going inside the nucleus, there is a large number of downstream targets that could be affected by this. So one thing that we're working on now is trying to understand what are the targets, or either primary, secondary targets of hERG-MP function in the cardiomyocyte inside the nucleus. What are some of the mechanisms that control when and how it gets inside the nucleus? And are there disease-specific variants for hERG-MP, the nuclear polypeptide, that we can identify and then map out a potential mechanism of disease? So from our standpoint, being electrophysiology lab, we wanted to do the EPhys. That's what we like to do. And so we decided that we would just start going through the list: IK1, IKS, ITO, calcium current, sodium current. So we started with IK1, and we haven't gotten past that because it's already turned into its own story, and we're trying to figure out what's going on there. So this has been head up by a research faculty in my lab, Eric Jimenez Vasquez. And so he started this project with a very simple premise, much like what we did with hERG1A in HEK cells. He used HEK cells stably expressing inward rectifiers. So he started with KR2.1. He's got GFP here. He's got wild type distal and then the delta NLS. He expresses them together and not the biggest effect. It looks like maybe the delta NLS is actually increasing the inward currents here, but not really sure. He does 2.3, 2.1 and 2.3 are both expressed in cardiomyocytes. Now it kind of switches a little bit. We're not really sure what's going on. But then he does 2.1 and 2.3 together, which is what you're going to see with varying degrees of stoichiometry, depending on whether you're in the atria or the ventricles. So these cells are expressing 2.1 and 2.3. He then overexpresses wild type distal, and he gets just an enormous increase in the magnitude of the inward IK1 current. He showed me that first, and I was like, wow, that's great. That's really exciting. But then he shows me that it also happens with the delta NLS. And I was like, damn it. What does that mean? Because we had this very clean idea of what the distal polypeptide was doing, and that it was dependent on going inside the nucleus. That kind of blew up our question, our hypothesis from the get-go. But it could also mean that maybe the C terminus of hERG, that distal C terminus, interacts with Kr 2.1, 2.3 heteromers as a macromolecular complex. So had we indirectly stumbled across a new, dimerization, or whatever you want to call that, between two different families of channel. So his next step was, okay, well, let's add in hERG1A and see what happens. So here he goes. He has 2.1 again. He overexpresses hERG1A. The currents get bigger. He adds in hERG1MP, and there's no additive change on the inward current. So whatever's happening on the inward side, hERG-NP has no additive effect. So hERG1A is either masking the interaction site, probably. And he repeats that for 2.3. He sees the same thing. Things get a little bit murky with the heteromers. So at this point, we're thinking, okay, it is just when we add in hERG1A, things get bigger, at least with the homomeric 2.3 and 2.1. That was somewhat supported by adding in hERG1MP. But when we dove a little bit deeper, we also noticed in the same cells, if we look at the hERG current, the nuclear polypeptide was disrupting the hERG current. And hERG-MP is making the hERG current smaller while also enhancing the size of the IK1. So we're kind of in this realm of uncertainty right now, right? We have similar trends across all three here, where it could be that hERG-NP has a specific effect on dual mode activity, where it's affecting ion channels both inside and outside the nucleus, or this could be an artifact we're picking up of something that is specific to the full-length channel. Now, all of that had been done in an HEK cell, so we wondered, can we reproduce some of these effects in stem cell-derived cardiomyocytes, where you have a more natural or physiologically relevant complement of ion channels? And I know, so IK1 and cardiomyocytes, you might be thinking, but Dave, you just spent like half of a seminar talking about how stem cell-derived cardiomyocytes don't have IK1, which is true. It's still there a little bit in the embryonic, but we have tricks that allow us to upregulate that, right? So if we did this experiment in an embryonic cardiomyocyte, yeah, you would be completely right. There's almost no IK1 here, and we wouldn't have the resolution to measure an effect on that. But we have lots of tricks to push them down that maturation pathway. And one of them that Eric is very good at is this micropatterning technique. So what he does is he coats a soft gel, PDMS. It's sort of like overcooked Jell-O jigglers. I mean, it's pretty soft, but it's still kind of like stiff to it. And then he coats it in a tight pattern so that the cardiomyocytes can only sit down and adhere in a very specific morphology. And it's this rod-like morphology, which is what you see from adult cardiomyocytes. So what you're doing is you're forcing the cell to grow in a way that will optimize the alignment of the contractile machinery. You're giving it a soft substrate so it can contract more physiologically, something that's more similar to what's happening in the developing myocardium. And when he does this, he gets a much more adult-like phenotype. He gets very clean alignment of his sarcomeric structure. And when he does patch clamp on the ventricles, the majority of these cells can be quiescent. You can see his injection current right here. He's got a very negative resting membrane potential. So we've got something that's much more similar to adult physiology. It's probably more like a newborn. It's still not full adult, but it's down that path. And you're not going to get this level of hyperpolarization of the resting membrane potential unless you have IK1. So, but then we ran into a problem. So we overexpressed the polypeptide in our immature and our matured cardiomyocytes, so the non-micropattern and our micropattern cardiomyocytes. And we stumbled across something where the distal construct only went in the nucleus in our immature cells. And I know that the sample size is really small here, but we actually did this experiment after patching a boatload of cells and seeing it over and over again. So we're like going back and quantifying what we had seen a million times under the scope with the patching. So something about maturing the cardiomyocytes prevents them from going inside the nucleus. Okay, so right now we're thinking that might be like a phosphorylation status or something like that. So you can see here, just like in the HEK cells, it's only overlapping with the DAPI stain. In these matured cardiomyocytes, these micropattern cells, you might be able to see a little immunofluorescence there, but for the most part, it's just packed into the SR and it's not going anywhere past there. But when we look at the effects on currents, if we look at the native IK1, GFP, we get some reasonably good currents. He adds in the distal construct, and those currents are roughly doubled. But then here with the IKR, he sees no effect. And that's actually really encouraging. We're piecing apart or teasing apart in the cardiomyocytes the effects that we'd seen in the HEK cells, where we're preventing the distal construct from going inside the nucleus. We correspondingly see no effect on IKR magnitude, whereas the distal construct sitting in the ER, and we're getting a corresponding increase in the magnitude of that current. So we think that these are two very separate effects. One is truly dependent on going inside the nucleus. One happens outside of the nucleus. It happens at the ER, maybe as a chaperone effect, promoting forward trafficking of the inward rectifier potassium channels. And Eric's doing a number of experiments to figure that out. But I mean, to summarize, HERG-MP seems to, it increases the magnitude of IK1. This effect is independent of the nucleus, but we haven't really ironed out whether this is dual activity from the HERG-1-MP itself, or if this is something that is really can be tracked back to a macromolecular complex between HERG-1A and the inward rectifiers KR 2.1 and 2.3. So we're working to figure that out. With that, this is my team. Abhilasha is our lab manager. She does all the cell culture and differentiation. I talked a lot about Eric's work today. He's a really phenomenal electrophysiologist, and I think he'll run his own lab one day. I didn't talk much about Francisco's work. He did some contribution in some of the electrophysiology. Most of his stuff is in the variants associated with sudden unexplained death and epilepsy, so that's fine. We have an army of undergrads that's always helping us out, and I always need to acknowledge our funding sources and be happy to take some questions now.
Thanks, Dave. Great talk. Thank you very much. Yeah, we have questions coming in. So the first question from Mark Rogers: Does importazole renormalize the functional hERG current profile of the distal C-term mutant, thus proving that nuclear entry is required?
That's really interesting. We never did that experiment. We were pretty well convinced when we did the delta NLS,
Mhm.
right? So with the delta NLS polypeptide, it doesn't go inside the nucleus, and we don't get effect on the surface membrane. But we could do, yeah, we could do the importazole. Um, we could try that. One challenge we did find is that with the importazole, at least in the HEK cells, is that we had to incubate for a very long time to diminish the polypeptide inside the nucleus. So we think that my guess is that we're missing a piece of the protein that might have an export signal or some machinery that would draw that down, because it seems to be a lot more stable in the nucleus of the HEK cell than it is in the cardiomyocyte. But that's an interesting idea, and it's something that we could apply to um, the immature cardiomyocytes as well to kind of piece that apart. There might be some challenges with off-target effects, but I'm sure there's a way to control it. It's a great question. It's cool.
Sure. Second question from Susie Kohout. Very cool talk. Did you look for a nuclear export signal? It would be interesting if the hERG nuclear gene gets shuttled out as well, or is it a one-way street?
Yeah, I screened it for an export signal when we first found it. I haven't done it recently to see if someone's got a better predictor tool, and we didn't see any hits initially. The predictor tools for export signals are far less reliable, though, than for the import signals. So that can be a little hit and miss. I've had a lot of conversations with Lori Isom's group, because they have a similar issue with a beta-1 subunit polypeptide that goes in and out of the nucleus, and it's challenging to figure out what the export machinery is. It's easier to find the import machinery.
Sure. Another question, anonymous question. Since you talked about KIR 2.1, did you try KIR 2.2 in the expression studies?
No, and for the only reason because we didn't have a stable cell line to do it in. We could do that if we had that cell line. We were just sort of using what we had on hand for that experiment.
Sounds good. Next question from Amanda Klein. Thanks for the great talk, Dave. Do you suspect that hERG may have effects on other inward rectifiers, like GERK and KT?
Yeah, it opens up a lot of questions. I think that we're going to have a lot of effects. So this is these are HEK cells, blank HEK cells, not expressing full-length hERG, and we can pick up a very faint signal that is similar to the hERG-MP signal that we see in cardiomyocytes. We can also see it in pancreatic cancer cells. Here's the signal in human iPS cells. We've seen it in immature neurons that we differentiate. So we think that from the standpoint of hERG-1MP, it's going to have a lot of effects on a lot of different things, and mapping out what it does exactly will take a long time. As far as the interaction with assumedly 1A, or well, hERG-1, the full-length channel, and inward rectifiers, our guess is that it's a coiled coil domain in that distal C terminus that's mediating this effect. There is one predicted to be in there, and there's a number of disease-causing variants that land in there well. So something like that has the capacity to have a lot of interactors. So my guess is that if it interacts with I mean, since it's interacting with, or seems to be interacting with 2.1 and 2.3, there's probably a number of other potential partners that you could see that might be tissue specific, moving into neuron or pancreas, et cetera, where the full length is also seen.
Sure. Next question from Artem Kondratsky. Thanks for your talk. I noticed that UIKR currents on your last slides are quite small. Some researchers use cesium instead of potassium to increase her current amplitude, reporting up to a tenfold increase. Have you tried this approach?
Yeah, I've used cesium in the past, and one challenge that I'm not always crazy about with cesium is that we do see a change in some of the kinetics of the channel. We see a change in particularly deactivation. So, I mean, usually for most of the stuff that we're doing, once we get into the cardiomyocytes, we're fairly comfortable with recording behaviors down to about half a picoamp per picofarad as long as the cells are large enough. So, but the cesium is not the worst idea for us if we're trying things that require a tighter or a cleaner signal to noise ratio. So it's not something we rule out. It's just that a lot of the stuff we're doing is a little bit more broad stroke, I guess. And then we also tend to use the time course of deactivation as a marker for channel composition. It's not perfect, but it can be an indicator of the relative abundance of 1A to 1B subunits in the tissue. And I don't know how cesium affects that, so I've been hesitant to try that. But cesium is a good tool.
Sure. Another question, do you know in which types of neurons are hERG channels expressed? Do you think the effect of hERG in the nucleus can be a nuclear transcription factor for another target gene as a beta subunit or peptide of hERG?
So we're working under the hypothesis that hERG NP is a transcription factor. We're going to set up and do some ChIP-seq and see if we can find specific binding partners from the DNA and get a cleaner map there. We've done some RNA-seq from iPS cells, and we can see a healthy change in gene expression there. Now, as far as what neurons is hERG expressed in, you could almost say all of them, but it's not like which ones are they expressed on. It's more like which ones are they upregulated. And we see them, a lot of hERG in pyramidal neurons. We see a lot of them in excitatory and inhibitory neurons in the stems, in the brainstem. We also see them in the hippocampus. The challenge, I think, for pinning exactly what hERG does in the brain is that there's also two other hERG orthologs that are expressed there. So there's 1, 2, and 3 in the human brain, and their expression profiles overlap a lot, particularly in the in the cortex and the hippocampus. So you can't do basic pharmacological drug subtraction or anything like that because their pharmacology is far too similar to separate them out. And there's only been one study that started to employ, some gene editing technology to get at that. That's something we'd like to do with some of our casein H2 null and gene edited stem cell lines to see how the selective contribution of hERG varies across different neuronal subtypes. The technology is not great, I guess, to do that with regards to differentiation, but you can get started on it. But it tends to be a lot in the brainstem, the pyramidal cells, and layer 4 and 5 seem to be somewhat enriched, and then you do see a number of cells within the hippocampus. But it's no like very limited population where you can say, oh yeah, it's gone from this population of interneurons, and that's how it causes epilepsy. It's unfortunately not that simple.
Sure. Since we are over time, well, very quickly, we have one question and one raised hand. So let's go with the question first. We observed from Elisabetta Kerby, apologies if I didn't pronounce the name correctly. We observed her very early in undifferentiated human embryonic stem cells and IKR as well, stem cell 2007. Also, HCN is present at the time. Have you ever tested the effect on HCN or plan to do so?
To test the effect of HERG-P on HCN, is that?
Um, I think that's
Yeah,
my
I
understanding.
mean, the short answer is yes. I mean, we'd like to test to see if HERG-P affects all of the channels. I mean, it was meant to be a quick scanning of the effect on the major the major currents of the cardiomyocyte. And we've run into a couple of, like, I guess you could say, like, we didn't expect that the distal CT wouldn't go inside the nucleus of the matured cardiomyocytes, for example. So we've started doing some workarounds there to more specifically target things in the native system, and we're getting the HEK cell lines that we need to go after those other currents.
Thank you again for sharing with us your very cool research, and and thanks everyone for attending, and looking forward to the next month's talk. I believe will be by Tamer Gemaluddin from the University of Washington. So thank you very much.